The availability of culturable model organisms has been fundamental to the advancement in many fields of study. This protocol, to our knowledge, is the only approach available for culturing Doliolids. Reliable Doliolid culture technology provides a unique experimental model to study organisms that are important in some of the most productive regions of the ocean and that represent the earliest evolutionary ancestors of vertebrates.
This technique is unique as it keeps the algal food particles and the organisms suspended in the water column without contact with the walls of the container as they experience in nature. This culturing method can be applied to the culturing of other small gelatinous and crustacean zooplankton species.
Because Doliolids have a complex life cycle, understanding and recognizing the needs of each life stage is critical. Learning the life cycle and how to recognize and handle them takes a lot of practice. In our experience, the only way to learn this culturing technique is to see it and do it.
Several other project participants, including Aaron Arneson, Nicholas Castellane, Natalia Lopez, Lauren Lamboli, Brianna Pierce, Aria Rodriguez Santiago, and Terrell Scarborough, also appeared briefly in this video.
Before establishing and rearing Doliolid cultures in the laboratory, clean and sterilize 1.9 and 3.8 liter glass culture jars by immersing them in the sodium hydroxide potassium permanganate solution. Allow the jars to soak overnight. Then transfer the jars into the sodium bisulfite solution. Allow the jars to soak overnight. Next, rinse the jars thoroughly with deionized water. Allow the jars to dry.